Hello, everybody. My name is Nicole Butler, and this is my presentation on our cure research project on how somatostatin receptor 2, or SSTR2, may act as a shared pain pathway across different lupus subtypes, and basically how it's a convergent pain pathway in lupus associated with chronic pain. So, this is a presentation by me, and it's supposed to be with my other group mates, Reagan Hopes and Samantha Anderson. All right, so first things first, we're going to talk about our background on chronic pain in lupus. So, chronic pain is basically a major symptom in various autoimmune diseases. Specifically for lupus, lupus has multiple subtypes, but they often damage the same organ, especially the kidneys in our body. So this just basically suggests that inflammatory and pain pathways across lupus variants, and then SSTR2 is a receptor involved in immune signaling and pain. So I just really chose specifically to talk about lupus, the autoimmune disease lupus, and talking about the chronic pain because I've had and have family members who suffer from this disease, and just seeing their daily challenges and struggles, especially with chronic pain, inspired me and really made me want to deep dive and learn more and understand the biology behind what they experience and why. So as I began researching, I was surprised to learn that there are multiple types of lupuses. I didn't know that there were multiple types. I researched there's a type called systemic lupus, and different other ones like the overlap syndrome. So even though it's these multiple subtypes affect different parts of the body, they most of the subtypes still have similar kinds of organ pain, especially in the kidneys. So the one question that sparked the project or sparked my interest, I want to understand why different lupuses subtypes can still attack the same organ and produce the similar pain patterns, even though they're different types of lupuses. So this led me to explore SSTR2, a receptor involved in inflammation and the pain signaling. So this project allowed me to combine my personal motivation with scientific curiosity to explore the potential pain and the potential shared pain pathway in lupus. So you're probably wondering why SSTR2. So SSTR2 regulates inflammatory cytokines, immune cell activity. So SSTR2 is basically important because it's involved in both inflammation and pain. So when tissues are inflamed for a long period of time, SSTR2 levels begin to rise, which may be why different types of lupuses causes similar organ pain, but it's basically pain signaling. So it's linked to neuropathic pain, and it could basically explain why I shared organ pain in different types of lupus. So that's why SSR2. So our research question asks whether SSTR2 is activated the same way in kidney tissues across different types of lupus subtypes. So if yes, SSTR2 could be a shared therapeutic target, but if no, then that's another question we have as well. So basically, do different lupus subtypes upregulate SSTR2 in the kidneys at the similar levels? And we have a little picture right here on how lupus affects different parts of your body. So if you can see, you can have the mouth, you can get mouth sores, chest pain, just different things like that. So this is my research question. And then with this research, you have to have experimental groups because it wouldn't be any research, or really wouldn't be a good research without experiment. So the type of experiment I was putting on is a 3-group comparative experimental study or a case control design, and basically where we're going to compare kidney tissues from 2 lupus groups and 1 healthy control group. So the healthy kidney tissue controls is a kidney tissue from a person without lupus. So the purpose for this is to provide a baseline for normal cytokine levels and normal SSTR2 expression. And then our 2 different lupus subtypes, we have lupus subtype A, which is SLE with nephris, and lupus subtype B, and it's another form of lupus with kidney involvement. So basically, these both going to allow us to see whether different lupus subtypes activate the same pathway, and um, allows us to see and test which SSTR2 upregulation are the shared cause of inflammation for chronic pain across the different lupus subtypes. So the different methods. So our methods, we use enzyme linked. This was a big word. Enzyme linked immunosorbent assay. It's basically a lab method to measure the amount of specific proteins, in this case inflammation cytokines, in tissue samples. So it works like a molecular magnet. So the antibodies in the test grab onto the cytokines, and we're looking for the cytokines that we're looking for, and an enzyme like basically makes a color. So the more darker, or the more present a color is, it helps to see how much inflammation each lupus subtype is produced. So that's the aim number one, or the method. And then another one, we use PCR. It's like basically to measure the SSTR2 gene expression and confirm like the protein level. So we then look at whether the SSTR2 rise together, and then yes. So aim one, focus on inflammatory side of lupus, while aim two measures the SSTR2 at both the gene and the protein level. And then aim three is combination of the first two aims, where we look at whether the cytokines and SSTR2 increase together, and if inflammation rises the same time as SSTR2 increases. So basically it's going to suggest that SSTR2 is a part of the pathway connecting to inflammation to chronic pain. So basically, these aims just help us understand if SSTR2 could be shared through therapeutic target across different types of lupus subtypes. So our quick methods we have is qPCR. It quantifies gene expression, how much mRNA is present, and then I keep calling it ELSA, but I know it's not. ELISA measures protein levels, so cytokines, chemokines, SSTR2. So our expected outcome, so we expect that lupus subtypes will show similar increases in inflammatory cytokines in the kidney. We also predict that SSTR2 expression will rise in both groups, which could explain why both subtypes produces the same kind of organ pain. And if cytokine levels and SSR2 levels rise together, that suggests SSTR2 may serve as a shared pathway behind the lupus-related chronic pain. So the significance in this research is that SSTR2 might be a shared marker of pain across different types of lupus. So if we know that SSTR2 rises constantly, we could use it as a diagnostic indicator for organ pain, or even develop therapies that target SSTR2 directly. So our student takeaways, basically RNA, um, oh, RNA extraction, pain pathway interpretation, and scientific communication. Here are my references that I used also in the essay, the prompt, or the abstract that we wrote as well. So these are scientific references used to support the research design, and they cover autoimmune pain mechanisms and the biology of SSTR2 reception to inflammatory signaling pathways, and how pain is present in lupus. So thank you. Hopefully this was